Hey all, if you're watching this, it's because you need more time working with the idea of meiosis or because this is your assignment for a remote day. I think for a bunch of you that might be the case. We went through some of this in class already for those of you who were there, and then so you're welcome to skip ahead to where you remember ending on your notes. If you are someone who wasn't here for this, then you're getting the lecture just on this weird format in a video. In a different way and you have time for it. Remember that parts of this content are on your test. The test isn't too hard, but make sure you look at your study guide. You all have it. It's also posted on the Google Classroom stream if for some reason you need access to it again. And then all of you should have a meiosis notes sheet so that you can take notes on this as you go along. I'd like them handwritten. I think you're just more likely to remember if you're like drawing and working through it. And yeah, I guess that's it. So go ahead and enjoy this video. So you saw the objectives on the last slide. You're going to learn a ton about meiosis. Please make sure to take notes on it in the note sheet. I think I attached the note sheet to the Google Stream as well. You could always email me if you're missing it. I'll be in full contact on Monday when you're working on this. But some big questions that meiosis is going to discuss and talk about. So where did you get your DNA or chromosome pairs from? Like if you think about it, your body has 23 pairs of chromosomes. Where did they come from? And why are they pairs? And are they the same? Are they different? Like what is going on? How many chromosomes total do we have? We have 46 total because you have 23 pairs. How many pairs do we have? 23. So you've heard of 23andMe. That's referring to your 23 pairs of chromosomes. Each of us another thing meiosis is going to reference or talk about is each of us has made, or we make, or has made, depending on your gonads, particularly, gametes. How many chromosomes do each of these gametes have in particular? And so we're going to get into that with this whole thing. So you can see on this slide, and this isn't on your test, I'm not going to ask you how many chromosomes or how many pairs of chromosomes you have, but I do want you to take a moment to look at this. So if you look and see, and on this, you'll notice there's no room for this on your note sheet. You want to take this in your own separate notebook. There's some information that we have to talk about before we get into the actual process of meiosis. But so go ahead and take this on a separate notebook sheet. And then you'll eventually, when we get to the process of meiosis, be on your note sheet. So, and there could be room on the back of your meiosis note sheet too. I don't remember. So all but one of these, if you look at this, this is your human chromosomes, and you have 23 pairs, like we talked about. So you look at number 1, number 2, number 3. The reason they have different coloring is highlighting different sections of DNA and what it codes for. You'll notice that each of them, each pair is responsible for coding different kinds of DNA. So it could be that on number 7, if you take a moment to look at that, maybe that colors that green band that's kind of hidden in there. Maybe that's like melanin amount. So it's like hair color and skin color and eye color. It doesn't have to all be on one chromosome, but we'll pretend like that's the case. Well, one of those, so the one on the left for number 7, we'll say is from your mom. That's the chromosome from your mom or from whoever had ovaries in your family. And they're going to give you maybe blonde hair color on number 7. And then the other one is going to be from whoever had testicles, whoever gave you the sperm to become who you were. And so that green band on that one, maybe they gave you a brunette section in particular. So that's why you end up with 2 different sets. And we'll get more into that when we talk about Mendelian genetics. But you can see one chromosome is from one parent, one chromosome is from the other parent. Most of these are homologous or relatively identical. They're not perfect, right? They're going to give you different options depending on which parent it came from. But number 1 and number 1 look almost the same. Number 2 and number 2 look mostly identical. 3 and 3 is the same. 4 and 4 is the same. But you'll notice that there is one set in here that doesn't necessarily look identical. Go ahead and see if you can find it. The help sits at the bottom right-hand corner. At least I believe that's where it'll be. See the XY? So this person, if we're looking at all their DNA, we're assuming that they have testicles because they have a Y chromosome. You'll notice the X and Y look very different. So that set is not identical. And I think most of you know that that set in particular is responsible for determining hormones and gonadal development. So an XY usually makes testicles and lots of testosterone. If it was XX, you would notice that those two X's are going to look very similar, and that's going to be responsible for a ton more estrogen, progesterone, and also ovaries in particular. So as always in biology, we're going to use a million terms because that's how biology works. All science does. It's got its own language, but I feel like bio in particular is a little crazy face with the terms. But we need to know the difference between diploid and haploid, and we'll get to haploid next. But diploid, that term you want to write down as a definition because we'll talk about this. So we each have copies of certain chromosomes, right? Not just one of each. That's what you saw on the last slide. We have two sets of chromosomes. So our cells that have two sets of chromosomes, which are most of the cells in your body, your cheek cells, your neurons, your hair cells, really your fingernail cells, your teeth cells, your tongue cells, whatever, they are called diploid cells. And they have a full set of DNA, which means they have two chromosomes. So two pairs of chromosomes per type of chromosome, like you saw in that last picture. So that'd be diploid. It's often drawn as 2N, as you can see here. So I feel like we've already talked about this a ton in class, but you know that there's some cells in your body, and I'd like you to think about this for a minute. Which ones, and these snails are giving you a hint. Slightly pornographic, I apologize. But what cells in your body are not diploids? What cells in your body do not have 2N? They don't have both sets of chromosomes. They have half the amount of chromosomes. So if you normally have 46 chromosomes, and some of your cells have half the amount, how many chromosomes do they have? 23, right? And why is it that these cells only have this many? So hopefully in that last slide you came up with like, oh yeah, sperm and egg cells, right? So all the cells in your body need a full set of DNA to run and function normally. But you do have these specialized cells that are just for reproduction that are called haploid cells, or you can call them gametes, or you can call them sperm and eggs. But those are the ones that are haploid. Those are the ones that don't have the normal amount of DNA. And the reason they don't is because we are sexually reproducing organisms. If it were asexual, that would be different. You would just essentially make a copy of yourself and move on. But with these guys, what we end up doing, and I think you know that in sexual reproduction, the egg and the sperm come together. Half the DNA is in the sperm, half the DNA is in the egg. The two together make a complete set in fertilization. But humans carry a half set in their gametes. So if you have ovaries, you have eggs. If you have testicles, you have sperm. And both of them are called haploid. And it kind of sounds like half, which is kind of nice, or haploid, but it's haploid, and it's referring to having only one set of each chromosome. And that's really, when we talk about meiosis, we're talking about the process of making these guys. The other ones, the diploid cells, making those is the process of mitosis. And remember, when we get to our test and you're trying to figure out the difference, if you're making sperm or eggs, it's meiosis. Anything else, tumors, hair growth, development, fetuses forming, all of that is going to be mitosis. So I actually brought up this term a little bit ago, but it wasn't explicitly on there. So when we talk about chromosomes that match from that picture we were looking at, so 1 and 1 looked very similar, even though one was from your mom and one was from your dad. So we call those homologous chromosomes, which means they have the same function, they have the same kind of DNA. They're not identical because this is going to have one set of genes, this is going to have another. Both will maybe determine how long your nose is. This one's saying a short nose, this one's saying a long nose. So we refer to those as homologous chromosomes. So if it's chromosome 1, regardless of what parent it came from, it's a homologous chromosome, it's chromosome 1. You'll also hear them called homologs, but they have the same genes in roughly the same location. So they're essentially the same kind of chromosome. So the general overview of meiosis, which we're about to get into, I know so much introduction, is that gametes are made via meiosis, which is a cell division process that is similar to mitosis but results in 4 haploid cells. And we've talked about this in class a little bit already, and it goes through 2 rounds of cell division. Because remember, cells can't split into 4 cells. They have to split just into 2. So if you go from 1 cell to 4 cells, then you have 2 rounds of splitting in there, and we'll work through this. But you'll notice that the initial cell actually has a full set of DNA, but the last cells, you see the ends down the side, they only have half the amount of DNA, and they're what are your sperm or your egg at the end of it. So if you haven't, you want to pull out your note sheet at this point because you're going to want to look at it for a moment. It's a lot longer than mitosis, and that's because meiosis is a longer process since it goes through 2 cell division cycles. They're distinct. That's similar to mitosis, which is why it's nice to start with mitosis and go into meiosis. And again, the whole result is making sperm or egg. There's 2 phases of meiosis, meiosis 1 and meiosis 2. You'll notice that like prophase 1 is labeled prophase 1, prophase 2 is labeled prophase 2, anaphase 1 is labeled anaphase 1, anaphase 2 is labeled anaphase 2. And they're really similar, but what you're going to notice is that what's going on with the chromosomes is different, and that's how you know the difference between the 2. And this is a really cool process, and this process also explains why you and your siblings look different, which is pretty interesting because of something important that happens in meiosis, which unfortunately can lead to genetic disorders as well, but specifically explains siblings and siblings looking different. All right, so let's start with interphase. So on your note sheet, you should find interphase. So before meiosis 1 can occur, which eventually will lead to mitosis 2, the diploid phase must go through an interphase. And remember, interphase, all this means is it's a growth phase, is what it is. Mostly. It's a growth and preparation phase for cell division. But this is when the cell is just existing as a normal cell. In meiosis, because we're going through a process of differentiation, you actually have a parental stem cell. So it's this interesting cell that doesn't look like sperm and doesn't look like egg and has a full amount of DNA that can make haploids, which is a pretty phenomenal thing to have. If something happened to these stem cells, you wouldn't make egg and sperm. You would still survive. You'd be fine. You just couldn't make offspring. But this stem cell in particular will differentiate and make different kinds of cells at the end of meiosis. So the stem cell during interphase, you should write it grows, it makes organelles. During this, it has its own like S phase, like we talked about in mitosis, where it replicates its set of DNA in preparation to go into meiosis, making the cell foreign for a good amount of time, a little bit of time, and then it starts developing necessary parts needed for meiosis, like spindles or centrioles, right? So to make the centrosomes. So all the little organelles that help, that you've seen before in mitosis, are also used in meiosis. So still in meiosis 1, but now in prophase 1. And remember, prophase is kind of a long phase in mitosis as well. This one is a little longer, and there's a really important process that happens in here. Normally, I would have you guys make this out of clay, and if you were in class, you probably did work through this and try and work it out of clay. But really, we'll get to that in a moment, but there's a really important thing that happens in prophase. I mean, prophase is always important. You can't move on without it, but there's this really cool thing that happens that explains why you look different than your siblings in this phase. So again, this is prophase 1 because it's part of that first cell division process in meiosis, in which there are 2. So in this one, you can start to, just like in mitosis, you can start to see the sister chromatids, right? You see the condensation of the DNA. It doesn't just look like an empty blur of a nucleus. You start to see distinct chromatids. And remember, we talked about this before. Sister chromatids are when you have X's. That means that you've duplicated the DNA and they're still stuck together. So if this is the original chromosome 1 from your mom, it's made a copy and they're still stuck together at this point. And that's what makes that X. You can see the distinct X shape for the most part, or at least see condensation of the DNA. During prophase, the spindle fibers form and reach towards the centromeres, right? They're not there yet. It's not quite moved on, but you see spindle fiber evidence. You start to see those centrosomes on the outside, the poles, to like designate which part is which side of the cell at this point. The nuclear envelope has disappeared because it would be problematic to have a nucleus intact and still allow for this at this point, which means these cells are more exposed at this point because their DNA is not protected. And then the nucleolus has disappeared just like in mitosis. So all of those are happening. We'll get to the next important thing on the next one. It's starting to hint at it up in the top right corner. Those are your first steps for phase 1. We're not done, but we're almost there. Okay, So this is that cool thing that happens in prophase. And again, prophase is super long in mitosis as well, but really long in meiosis one. So I want you guys to all look to the upper right. Just take a moment to look at that. And again, if you were in class, you probably played with this already. But notice that really, if this is your mom's DNA and it's going to eventually make, say, eggs, right? So if you look at that, the red one on the left, that's chromosome 1. That's actually from your grandma. That's your grandma's DNA in your mom, right? That's the one she got from her mom, which would be your grandma. It's duplicated, which is why it's an X, right? So the one on the right, that's yellow, that one is from your grandpa, or it's specifically from her dad. It's from your mom's dad. So what happens during this process is those two, they're all chromosome 1, but they're her different copies, and they've been duplicated at this point. But all 4 chromosomes get next to each other and scoot next to each other and end up switching and swapping sections of their chromosomes, so sections of DNA. And what that ends up making at the end is it ends up making chromosomes that look different than your mom's chromosomes. So it could be, and these are resulting in eggs, those are eventually going to pull apart, right? Those sister chromatids, just like they did in mitosis. And you're going to end up with one line. Well, that line chromosome, that number 1, is now going to be a mix of your grandma and your grandpa's DNA. So it's no longer just from your grandma, and it's no longer just from your grandpa, because that's what your mom has. She has a chromosome from her mom, a chromosome from her dad. But what you will have is you will have some combination of her 2 chromosome pairs in 1 chromosome, which is kind of bananas. And you'll see that as we follow along. But that's why you and your siblings look different unless you're identical twins, is because you got a chromosome maybe with tons of grandpa DNA. Your sister got a chromosome with tons of grandma DNA. It's going to look very different depending on how different your grandparents looked. It could be that you and your siblings look very similar. And you're also going to get that from your dad as well. You're going to get a combination chromosome, because this is the same for spermatogenesis, or making sperm, a chromosome that has a combination of both his parents' DNA. And every single sperm is going to have a different combination in that chromosome, and every single egg is going to have a different combination in that chromosome. They never overlap and rearrange the same way. So it's really important that you get this down. It might be easiest to draw it and label it somehow, but I want you to note that these sister chromatids end up forming a tetrad where there's 4 of them touching each other, and they swap DNA, and you end up at the end of it with a different chromosome than what's in your mom's body, which is really interesting. Okay, so let's move on to metaphase 1, much easier step. So those sister chromatids that have been rearranged through the tetrad are now just X's again, right? They no longer are a group of 4 stuck together. And they do what you've seen before in mitosis, which is that they line up down the center. But you'll notice what's different from mitosis is with mitosis, it was an X and an X and an X all the way down the line. This actually, they're right next to each other. So those tetrads are actually right next. They're not touching anymore necessarily. This is showing them touching. But at this point, they're lined up down the center. And that rearranged chromosome 1 that's from your grandpa and that rearranged chromosome 1 that's from your grandma are right next to each other. And you'll notice that when they split, they're still going to be in X's. And that's going to be very different, or sister chromatids. And that's very different than mitosis. You'll notice the spindle fibers are now attached to the centromeres, which is the center of the chromosomes, but it's not going to rip apart those X's, and that's probably worth noting. It's not going to rip apart the X's, but it's actually going to pull them to either side, and that's very different than mitosis. So in anaphase, again, I would make sure to really draw, anaphase 1 in particular, I would make sure to draw that it's not that those chromosomes or those chromatids are splitting, right? That's what we saw in mitosis, where one would go to one side and one would go to the other. But in fact, you have 2, an X going this direction and an X going this direction. And so make sure you note that. But it's very similar to anaphase otherwise. It's going to kind of look like claws. But it really, they're still doubled chromatids. They're being pulled in opposite directions as opposed to be splitting apart at this point. So for telophase 1, this looks very similar to telophase and mitosis, which is convenient. It kind of looks like it's starting to, it doesn't have to look like it's going through cytokinesis. It could be like up in the right-hand corner, a nice full-looking cell where there's segregated DNA on either side. You're unfortunately, if we were looking at this under a microscope, you wouldn't really be able to tell right now, but you still have sister chromatids. They're still stuck together. So in your drawing, you should note that if we were looking under a microscope, it'd be hard to tell that at this point. But you can also see there's only 2 cells right now, and that's a big difference from when we look at telophase 2. There's not 2 cells. There's 1 cell with 2 sections, and that's different. And you'll see why that makes a difference when we look at telophase 2. But really at this point, all I want you to write down is that it's 1 cell, it has a little cleavage, or maybe it doesn't, but the DNA is starting to get segregated on either side of the cell. The nuclear envelope is starting to reform. The nucleolus would be coming back. The spindle fibers are disappearing. The centrosomes are maybe still there, but it looks like they're still stuck together, but it's starting to become 2 full cells again, just like in mitosis. But you might want to note that you still have X's, still sister chromatids. They haven't been pulled apart yet. So I hope you took a minute to think about that last slide. We've gone through cytokinesis with plants and animal cells before. There's just a slight difference. And this slide is just to be like, see, I promise, they make sperm, they make eggs. For some reason, a lot of people believe that plants reproduce asexually, and some can, for sure. And some can self-fertilize, that would still be sexual reproduction. But they do make pollen, which is sperm. So if you have allergies in the spring, it's often because plant sperm is getting into your nose. And they do make eggs. That are pollinated, right? So they go through their own version of sexual reproduction, but they make gametes as well. And so when we talk about the difference between cytokinesis, between plant and animal cells, please believe me that plant cells also need to go through meiosis, and they also need cytokinesis for this process. So this one's pretty straightforward. It looks just like mitosis. The only difference, which you actually can't see in this picture, it's not representing meiosis cytokinesis, is you're still going to have the sister chromatids, right? So it's going to look like 2 cells being pinched off for animal cells, right? You can end up with that contractile ring, that thing that kind of squishes the membrane so they're touching, and then you have 2 cells. As a reminder, for plant cells, the only difference is it needs to grow a cell plate in between as well. Remember, plant cells have membranes and cell walls, and that cell plate is finishing off the cell wall. So now at this point, you have 2 new daughter cells, each with 2 N chromosomes, so a full set of DNA, but it's all rearranged at this point. But we're not done, right? Because you need to end up with half the amount of DNA. Right now you have a full set of DNA, but we actually need to end up with half the amount of DNA. So go ahead and look at, I know this is obvious, but look at the picture. The far left cell is the first cell. The far right cells are where we are right now. What's different between the 2? Tell me 2 differences. Or mentally think about it. So 1, in the original cell, you have linear DNA, like most of your cells do, unless they're going through replication or division. In the new cells, they're in sister chromatids. But also you'll notice that the DNA is rearranged at this point. So when these separate, you're going to have 1 chromosome is going to have a little bit of grandpa DNA and grandma DNA. The other one's going to have a little bit of grandpa, a little bit of grandma. And it could be that you get no rearrangement as well. Most likely you will get some. Sometimes it's substantial rearrangement. Sometimes it's just a little bit of rearrangement. But again, this rearrangement is why every single offspring is going to have some genetic diversity and look different and distinct. So there's not a second interphase. Woohoo! You don't have to go through another interphase. Once meiosis 1 is done, once cytokinesis is done and it split those 2 cells that now have sister chromatids in it and a full set of DNA, you now head right into prophase and meiosis 2, which is awesome. So conveniently, meiosis 2 looks a lot like mitosis, which is nice. But you'll notice if you look at the upper right-hand picture, right, of meiosis going on, you're eventually going to end up with 4 cells. So what's going on and what's different is you have 2 cells going through this right now. That's why you have so many circles on your picture, because right now it's not just 1 cell going through it, it's 2 cells. But this is going to look very similar to mitosis for both of them, which is really nice. But in this phase, each sister chromatid, remember they're stuck together, becomes visible. So you start to see them, and you can see them up in the pictures as well. The nuclear envelope disappears, just like you've seen before in prophase. The nucleolus disappears. The spindle fibers are reforming or regathering or going where they belong. And then you're going to see some centrosomes as well starting to form, or poles. So make sure you draw that in both of them, because you have two going through it. Make sure you write those key points. Again, longest phase of this section as well. So in meiosis 2 and metaphase 2, so we're in the 2 section, right? Because we're in meiosis 2, so that should have been prophase 2 on the last one. What you're going to see now, again, it's going to look like metaphase and mitosis, but with rearranged sister chromatids. That would be the different. And I know I keep saying that, but that's really what's distinct about this. But you'll see the comparison in metaphase 1, where it was 2 X's next to each other, and the X's just got pulled off. I'll like make X's like that, and they got pulled either direction. But now in this metaphase, you actually have X above X above X, because they're going to be split in half. So you just have a line on either side, right? A single chromosome. So you'll see the spindle fibers are once again grabbing at those centromeres because it's going to rip them apart. And then the centrosomes are there, which are the poles on either side that help control and determine which side is which. And yeah, and they're lined up. But you have 2 cells doing this at this point. This question, how many cells are going through metaphase 1 and meiosis 1? You just have the 1, but how many are now going through it? You got the 2. So for anaphase 2, again, very similar to mitosis, but 2 cells with rearranged chromosomes are going through it. If you don't remember that, I don't know what to do. I mean, I am saying it so many times that maybe you will forget it because it sounds like background noise. But similar to anaphase and mitosis, the paired sister chromatids separate. These X's are pulled, so it's 1 chromosome going in either direction. So it's starting to segregate on either side. Remember, those spindle fibers are shrinking and pulling the chromosomes with it. Telophase 2 looks very similar to mitosis, but you have 2 cells going through it. So you may see like pinching, you may not. You're not going to look at this under a microscope anyways. But at this point, you have what looks like 2 full cells, right? With the nuclear envelope reforming, the spindle fibers going away, the centrioles going away, the nucleolus reforming, the DNA uncondensing at this point. So it's starting to look like 4 cells kind of stuck together still. In the picture we have over there, you see that dark green thing? It's the cell plate starting to form. So that's starting to get into cytokinesis. But when it still looks like it's still separate, uncondensed nucleuses with no division, that's when you're in telophase. Telophase and cytokinesis start to creep into each other. But make sure you get that down, and make sure you draw it out for all what look like there's like 2 cells stuck together and 2 cells stuck together. So cytokinesis is going to be the same as before. It's just that now you're making 4 cells. So you're going to divide this set and divide this set. So again, if it's an animal cell, it's just going to contract and pinch and separate, because of the cell membrane. When we played with bubbles and looked at it, like you can add on, you can take out chunks, you can cut the bubble and move it. So that's the way the cell membrane works. With the plant cell, you're just going to have to grow plates in there. So each one will have to grow a cell plate as well to encourage that cell wall to develop. I do want you to look at that like purplish picture in the middle. You'll know, right? You can barely see it. I should probably make it bigger somehow. But each of those chromosomes, so if those are representing, each of them is like a different egg or a different sperm. And so you'll notice that each one, even if that's chromosome 1 and chromosome 2 at this point, each one looks incredibly different. I feel like the people who drew it made it a little weird because it looks like it's both like 2 sets of chromosome 1 in each one, but say it's chromosome 1 and chromosome 2. It's got a little grandpa DNA, little grandma DNA, and each one is going to have a different combination. And that's because that recombination phase in the initial prophase, that tetrad that is mixing up the DNA. And that is why you and your siblings look different. It's because each sperm and each egg is going to have different amounts of grandma and grandpa DNA on each chromosome given to you from each parent. Okay, So let's talk a little bit about gametes. Now that you know how they're formed, right? Meiosis, again, makes sperm or eggs or gametes in general. Let's talk a little bit about human strategies for making them and how they produce them and how many they produce, how often they produce. Because it's interesting if you think about people with ovaries versus people with testicles, how they make gametes or how often. So when does meiosis or spermatogenesis, like the creation of sperm, happen in males? Puberty, whenever that starts for that human, on until like they're too old maybe to start making a lot of functional sperm. But that could be a long time. That could be 70, it could be 60, it could be 80. When do people with ovaries make their eggs? That actually happens in the womb and then never again, which is really interesting. You are born, if you have ovaries, with all of the eggs you will have your entire lifetime. And they're made at that point. And it's interesting, we talked about that in class. This starts going into the last part, which is advantages and disadvantages. But if you make them at a young age and say you then go stand in front of a nuclear power plant for 4 years of your life, you'll destroy all the eggs you made and you won't make any more. If you are someone who makes sperm and does that, in theory, as long as you don't mess up your stem cells, you could just make more sperm. You could make more functional sperm. If you have messed up sperm for 4 years, you could go back and make more. If you mess up those stem cells, that's different. But if something happens, you can just make more. And it's really interesting. So how often can biological males or people with testicles release their gametes? They can do that once they're in puberty, a lot, whenever, and it's an excessive amount, right? How often can people with ovaries release their gametes? It tends to be once a month. It depends particularly on that person. And then what are the advantages and disadvantages to either? Really, the advantage of making a lot of them often is if you think about a small group of organisms, so we can move away from humans to be less creepy now. So you have a small group of foxes, right? And you have a bunch of females and a couple males. If you're trying to grow this group, the males can inseminate whenever they need to so that the females can get pregnant and produce offspring as often as they are able to. Now, the females are the people with ovaries, the foxes with ovaries in this scenario. It's good for them. They can only have so many pups at a time. In their scenario, they're going to drop a few eggs. But for us, as humans, we tend to drop one. Sometimes you can drop a few more. And the advantage of that, not dropping them all the time and stopping actually once you're pregnant, is so that you don't get filled with babies and explode essentially. And also, pregnancy is hard on pretty much every organism. So it's good that they are strategic and timed at when they will reproduce so their body is not constantly being drained of resources. So the two are really interesting, but it has a lot to do with which ones ends up carrying the offspring and putting that investment, that energetic investment into it, and the one that is just adding their DNA to that gene pool. So meiosis is actually slightly different for people with ovaries versus people with testicles. And I want you to look at this. On the left, this diagram. On the left, you see meiosis for eggs, or oogenesis, right? On the right, you see meiosis for sperm, or spermatogenesis. Now look at the results at the bottom. Go ahead and skip down. The very end. You'll notice, if you look to the right first, look at the sperm. You know they're the sperm because they have the flagella, right? So one round of meiosis is going to make 4 viable sperm. And you know, we just talked about it, people with testicles, they make a lot of sperm all day, every day. If you go to the other side, though, and you look at oogenesis, right? And again, that happens in the womb before any person with ovaries is even born. They're born with all their eggs. They've gone through meiosis and oogenesis. If you look down, you'll notice that most of the cytoplasm and most of the organelles that we don't really see in this process, right, are actually segregated into just one of the 4 cells being produced. And the other 3 aren't functional eggs. So people with ovaries have far less gametes, and even their process of meiosis. You'll also notice, though, that the egg is much bigger, even though it's not showing it great in this picture, much bigger than the sperm. The egg actually ends up contributing the organelles, the mitochondria, so much of what ends up going into the final cell once it's fertilized to start making the embryo comes from the egg itself. The DNA, though, is split. That's really all the sperm is for, is it's to carry the DNA specifically to the egg. The egg actually needs to be a host, and it needs to have mitochondria, and it needs to have glucose, and it needs to have the things it needs to start cell division without any help from the womb for a while. So it's really important that it's large. And so meiosis for eggs tends to put all that stuff into one, and the other three are called polar bodies and just go away. So every round of meiosis, technically, for those of you with ovaries, only makes one egg, even though it could make 4. Just as a quick reminder, I just want to make sure this is all clear because a lot of people get confused at this point. Meiosis makes the gametes. Meiosis is the process responsible for ending up with these cells that have half the DNA, sperm or egg, right? Once fertilization happens, and fertilization is when the two come together to make something called a zygote, and that's a full set of DNA. It's the predecessor to the embryo. What happens is not meiosis at that point, but this one cell goes through mitosis and make 2 cells, and those split and make 4 cells, and those split and make 8 cells, and those split make 16, and those make 32. So rapid division happens at this point, but that's mitosis. So again, once that egg is fertilized, you are now going through mitosis again. Meiosis only results in sperm and egg. Everything else that involves cell division in your body, you're looking at mitosis. So you're going to see something like this on your test. So I'd like you, don't skip ahead, stay here, stay on this slide. Take a moment if you're flipping through it, right? If you're watching a video, you might want to pause it right now. I want you to try and answer these questions. If you can answer these questions with 100% accuracy, you're going to be set for this section of the test. It's not the exact same questions, but they're very similar because you know the difference between mitosis and meiosis if you can answer this with 100% accuracy. So go ahead and try. And then when you're done, so pause it maybe, try, pause it, give it a shot, answer them all either in your head or on a piece of paper, but commit. Don't say, I don't know. Just give your best guesses if you're taking the test. And then let the video go and look at the last slide and see how correct you were. If you were incorrect, think about why. That's how you learn, right? Processing why it was incorrect is going to help you understand what you got wrong. And if you got it all right, sick. Make sure that you did and think about why. And if you guessed on one, think about why it was correct. And that'll help you for your test. I promise.